Hi everyone, my name is Anna and I work in a pharmacology lab at Purdue where I test different properties of plants that contain substances known as opioids. The reason that I became interested in this research is because I really love plants and I find it so fascinating that many of the drugs that we use today, such as aspirin, several antibiotics like penicillin, and even some chemotherapy drugs come from plants or fungi. But besides these purified drugs that you can buy or are prescribed, there are many natural products or herbal supplements that are available for people to buy and use. And most of the time, these natural supplements can be helpful to the user, but if the product in question contains addictive substances such as opioids, problems may arise such as drug abuse. So again, in my research, I study the opioids that are present in these natural products and determine whether they're promising or problematic. So I'm sure that many of you have heard of opioids, such as morphine or fentanyl, which both have contributed to the infamous opioid epidemic in America. These opioids bind to opioid receptors, of which there are three: the mu, the delta, and the kappa opioid receptors. Many of the opioids you've probably heard of, like morphine and fentanyl and oxycodone, these all bind to the mu opioid receptor to create their pain relieving and addictive effects. When the opioids bind to the opioid receptor, they trigger different signaling pathways inside of the cell, where the receptor can signal through G proteins, leading to a decrease in cyclic AMP in the cell, which is, which is a very common messaging molecule inside of the cell, or the receptor, through a few extra steps, can also recruit another protein called beta-arrestin to lead to other different signaling pathways downstream. So you see that when an opioid binds to an opioid receptor, there's a few different things that can happen inside of the cell. Well, it gets even more interesting because recent research has indicated that different opioid structures can create unique signaling pathways after activating the opioid receptor. For instance, one opioid structure can force the receptor to signal only through the G protein pathway, or maybe the opposite. Maybe it forces the receptor to signal only through beta-arrestin. Of course, it could also be an unbalanced combination of the two of these signaling pathways. But regardless, think of the signaling pattern as a unique fingerprint to that opioid. And this unique fingerprint can help us find new drugs to treat pain relief and alcohol addiction, with fewer side effects that led to the widespread opioid epidemic. And so knowing this, I study different natural products that contain opioids to determine what their unique opioid signaling fingerprint looks like and whether the signaling profile contributes to negative side effects. The two plants that I study specifically are kratom, which is from Southeast Asia, and many people use this to self-treat drug addiction, and akawama, which are seeds from a tree in West Africa that people have used historically to treat pain. And so in my research with kratom, I've used cellular studies to determine that unique opioid signaling profile. And then I take those opioids and I look at them in mice. And for kratom, I used mouse models of alcohol addiction. And we actually found that the opioids that are in kratom can decrease alcohol drinking through their signaling at the delta opioid receptor specifically. In my research with akawama, I again used cellular experiments to look at that unique signaling profile of those akawama opioids, and then I tested them in mice to see if they did have, or if they did provide the pain relief that historically humans have used it for. And we found that although akawama does have opioids that bind to the mu opioid receptor, remember that's the same receptor where morphine binds, the akawama opioids aren't potent or strong enough to decrease pain in the mice, unfortunately. So to wrap up, my research on both akawama and kratom helps to better characterize the properties of the opioids that are present in these plants, and may provide guidance for their future regulation. Again, my name is Anna, and if you guys are interested in hearing more about my research or have any questions about research in general or higher education, please feel free to reach out. I would be happy to talk. Thanks.